Assalamu alaikum and welcome to the Dr. UUT channel. In this video we will talk about the cell cycle meiosis, somehow pronounced also as like meiosis. Well, before I proceed, I would love to mention that this video is available in Urdu, so now we'll commence the video in English. Meiosis. First, let us know the meaning of the word meiosis. Meiosis means reduction, means when there is a decrease in something. Well, this is about the chromosomal meiosis. So here what will happen? The number of chromosomes will decrease, the very first thing. How will it decrease? We'll know later now in the same video. And now coming to the point where this type of cell division occurs. Meiosis occurs in the germ cells. Now what are germ cells? The gonads, which are forming the male and female sex hormones like sperm and egg. So in order to produce sperms and eggs, what will happen? Meiosis will happen. That's it. Then coming to our next point, that it is a reduction type of cell division, and this the number of chromosomes will reduce. So generally, if I give you example of a human body, what we have the number of chromosomes are 46 in a human body. So now, in order to reduce the number of chromosomes, what is supposed to happen? One cell is supposed to undergo the division twice. Just like here, a diploid cell is there of a human body which contains 46 chromosomes. So this diploid cell will then divide into two daughter cells. And then what will happen? Again, these daughter cells will go in the second division, will form 1, 2, 3, 4, means two more further sub-daughter cells of the daughter cells. Parent cell will give birth to two daughter cells, and each daughter cell will give then birth to two more. Like this, one cell will produce 1, 2, 3, 4 daughter cells. So what will happen? The number of chromosomes present in the first cell, in the parent cells, are 46, and it is diploid. And this diploid numbers will be given to the daughter cells. And when the daughter cells are dividing, then they will produce haploid two sub-daughter cells. Now the question rises here that if this diploid cell is giving a two diploid cells, daughter cells, means if this diploid cells, this one is giving a birth to two daughter cells, and both daughter cells are having the same number of chromosomes, means both are diploid. Then if this daughter cell is giving birth to sub-daughter cells, then why the number of chromosomes in them are become reduced or halved? You can say a diploid will become diploid, then this diploid will become haploid. What is the reason behind and what is the logic behind? Very simple. In the first cell cycle, this will be just like mitosis we studied earlier. What will happen? The cell will enter in the interphase, and in the interphase, what we have phases? G1S and G2 phase. Suppose now this cell wants to duplicate itself or divide itself. So in that sense, the cell will enter in the interphase, and in the interphase we have G1S and G2 phase, just like mitosis. Then we have M phase. So, but in case of the meiosis, now we are studying the meiosis, the cell will enter into the G1 S phase, the replication of the chromosomes will occur. Then G2 phase will occur, the replication of the DNA. Okay, the cell will enter into G1 phase, then into the S phase, where we studied that these phases in a very best manner in mitosis. Now just I am giving you a little bit hint about these steps again. G1, and in the S phase the replication of the DNA happened, due to which the cell need now, because 46 chromosomes are not enough to distribute in each, so it has to be doubled. That's why what will happen? First of all, in the first cycle of the meiosis, cell will enter into the interphase, and in the S phase the replication of DNA will happen, and in the future then chromosomes will be doubled for the equal distribution in the daughter cells. Then in the G phase then will come the M phase. So in the M phase, meiosis will happen, and after the meiosis, then the cell will enter into the cytokinesis. Two daughter cells will be formed. then here the chromosomes that were supposed to be doubled by the DNA replication. So now we have 2 copies of the DNA. So the chromosomes produced in these 2 daughter cells will be equal. Means before we were 46 chromosomes, what we formed by this DNA. Now the DNA here is replicated, doubled. So at the end, what will happen? 92 chromosomes will be formed and will be distributed to the 2 daughter cells. And after the M1 means meiosis 1, there will be another cell division in case of meiosis. So we studied and we know about this, that meiosis occurs in 2 steps, in 2 phases, meiosis 1, meiosis 2. So meiosis 2 will start from the daughter cells. This daughter cell will then undergo in the process of cell division. So this daughter cell will enter directly into the M2 phase. In this phase, what will happen? The S phase is skipped. So means that this daughter cell has not undergone the process of DNA replication. So if DNA is not replicated, then the number of chromosomes will be the same that is present. The number of chromosomes will be the same that are present in the single cell. So now here 2 daughter cells were formed in the cytokinesis, and these cytokinesis daughter cells were diploid, and when one diploid cell will enter into this M2 cycle, and again the second one will also enter into the M2 means meiosis 2. So now daughter cell will undergo the process of meiosis. Means here what will happen? This meiosis is just like mitosis. This will then move to the cytokinesis. So now in this phase, what happened? You know that from cytokinesis to M2, then again cytokinesis, a phase is missing, that is known as interphase of the S, and the S phase is missing. So if the S phase is not available, means that the DNA is not replicated. So now DNA is not replicated, so the number of chromosomes will be the same. And when this undergoes the process of division, so when now this daughter cell is going to divide, so here the number of chromosomes are 46 and it is not duplicated. That's why when this daughter cell will undergo the process of the division, it will produce 2 haploid cells, and in each cell means there will be the half the number of chromosomes of this daughter cell. Now here what number of chromosomes? 46. So this daughter cell skipped the S phase. Now here is no S phase. That's why what happened? The 2n number of chromosomes were diploid here, means there were 46, and then 23 will be provided to each daughter cell, means each sub daughter cells by this daughter cell. So like this, the reason is clear that this daughter cell is not distributing the diploid number of chromosomes to the daughter cells, means to the sub daughter cells. So like this, what will happen? This parent cell will give birth to 2 daughter cells, diploid daughter cells, and this diploid daughter cell will then give birth to 2 more daughter cells but haploid. Reason is that S phase skipped. We got that. Now coming to the further explanations, how the haploid cells are produced in some biological explanation point of view. Well, we have 2 phases, M1, M2, meiosis 1 and meiosis 2. In the meiosis 1, we have prophase, metaphase, anaphase and telophase. After the interphase, we'll start the meiosis 1. So we got that number of chromosomes are doubled now. Now in this cell we have about 92 chromosomes, means it is a diploid cell, but now it has prepared itself, mean the number of chromosomes now it has gained now. Now before it, in this diploid cell, before there were the 46, because now it wants to divide, so for that it has also prepared itself in the interphase, means it has already gained now 46 more. So now this cell has 92 chromosomes. This will enter into the prophase 1. Now the story here of these phases is matching to the just like the phases of the mitosis, but a little bit difference is here. That is the reason, that is because of the mechanisms are happening here between the chromosomes. In the prophase 1, what will happen? In the prophase 1, homologous chromosomes will pair up in the bivalent and tetrad shape, means the homologous chromosomes will pair up. That is a very difference hereby. There were just alignment, the chromosomes were appearing in the prophase. Here they will be also appearing, but the chromosomes alignment will be there in the of the homologous chromosomes. In case of the prophase of the mitosis, there were just appearing of the chromosomes. And here the second thing that is difference is here the homologous chromosomes will appear and will pair up, and then after pairing they will form the synapses, and they will come after one other, means they will cross over each other. Like this, they will also change the segment of one another. Means segment of blue chromosome, example, blue chromosomes will be given to the black and the blacks will be given to the blue. Like this, they will change the segments. This segment changing will happen in the prophase 1. This is what making the prophase different from the prophase of the mitosis. So meiosis prophase 1, here what will happen? Homologous chromosomes will pair up and the crossing over will happen, and they will change the segments of the non-sister chromatids. Chromosome A will change, give its segment to the chromosome B, and chromosome B will give its segment to the chromosome A. Half of segments of the non-sister chromatids will be shared. And we have got the concepts of non-sister chromatids in our further videos if you don't know. If you know, it's fine. Well, then it will enter into the metaphase. In the metaphase, what will happen? The homologous chromosomes will come and will reach to the metaphase plate, which is the equatorial region. And after that, a kind of tension is created by the centrioles. These centrioles will try to move these chromosomes, homologous chromosomes aside. And in the anaphase, the chromosomes will be moved to the sides. Now here again, this anaphase 1, metaphase 1, prophase 1, metaphase 1, anaphase 1, telophase 1. Then we will go for the 2-1. Well, in the anaphase 1, what will happen? Now these homologous chromosomes will be pulled. And this phase is again different from the mitosis. Why? And what is the reason of difference here? The reason of difference is here. In the case of mitosis, there were the cohesive proteins present in between the sister chromatids. Those were broken, were degraded. Here in the anaphase, the proteins are intact, means proteins are available, the cohesive proteins are available. And now the complete chromosome will move. The sister chromatids will not separate. Like this, the chromosomes will move in the anaphase, and in the telophase, a cleavage furrow is formed by the actin filaments in between the cells, and which will pinch the cell inwards in forming the two cells. Cytokinesis will happen and two cells will be formed, and then what will happen? The chromosomes will decondense, and again two fresh cells are available. You can say daughter cells are available with the number of chromosome equal to that of the parent cell, means diploid. 46 here, 46 here. We can say the diploid cells are formed. Now this daughter diploid cell will enter to the meiosis 2. This is the meiosis 2. Now meiosis 2 is completely mitosis. Completely mitosis. How? Like in the prophase 2 of the meiosis 2, the cell will enter into the cell cycle, means again the centrioles will appear and the chromosomes will reappear. Here they were decondensed, the nuclear membrane will be vanished and chromosomes will be appeared, centrioles will move to the sides here and they will start producing the spindle fibers. And in the metaphase 2, the centrioles completed the spindle fiber formation, and they will hold the chromosome here like this, and they will start in the metaphase, they will start attention, they will start in the you can say the stretching of the chromosome. One centriole will try to move the chromosome to its side, another will try to move this chromosome to another side. Like this, the tension will increase, and in between, I told you before that in between the chromosomes, sister chromatids, there is a cohesive protein which is holding these chromosomes, means these sister chromatids, and that protein will degrade in the metaphase and in the anaphase. So what will happen? The centriole will succeed, will get the success by increasing. In the anaphase, the centrioles will carry the sister chromatid, or will separate the sister chromatids now. Here the homologous chromosomes were separated in the meiosis 1. In the meiosis 2, the sister chromatids will be separated, and in the telophase, what will happen? The chromosomes now available here are, you can say, half. Why? Because now this diploid cell had the 46 number of chromosome, so it missed the S phase and it entered into the prophase. And here we got the concept from cytokinesis, we got the M2 phase. From the M2 phase, we got cytokinesis again. So here is no S phase, number of chromosomes are 46. Now this 46 chromosomes will enter to the prophase, metaphase and anaphase. In the anaphase, what will happen? Half the chromosomes will be moved to one side and half the chromosomes will be moved to another side, means 23 distributed. In the telophase, the actin filaments will activate, actin ring will activate. This actin ring will pinch and will try to separate these cells, and in the end, 1n, you can write 1n or 23 chromosomes, two haploid sub-daughter cells are formed. So like this, the parent cell gave birth to the two diploid daughter cells, and the daughter cell then gave birth to two haploid daughter cells. The reason is that the parent cell went before the interphase and the S phase. Here, the daughter cell skipped the S phase, due to which the number of chromosomes were not doubled, or you can say DNA replication did not happen, due to which the chromosome became half, or were distributed half to the both the cells. Like this, the sperms and the eggs will be formed in millions of numbers. And like this, what will happen? Meiosis happened, means a diploid cell gave birth to two diploid cells, daughter cells. Those daughter cells then went in again the next type of division, the next cell division, and they gave birth to two more sub-daughter cells named as haploid cells. So like this, one cell produced four haploid cells. This is all about meiosis. Hope so you got. Thank you for watching.